Hello, and welcome to another episode of the Path of Longevity podcast. I'm your host, Dr. Stephen Sidoroff, and I'm joined by Dr. Rob Lufkin. Rob?
Hey, Steve, it's great to be here today. I'm so excited about this episode. We're joined by Dr. Lucia Aronica from Stanford University, who's a leading expert on DNA epigenetic methylation clocks. And specifically, her interest is in how we can program our epigenome with lifestyle factors. Hi, Lucia, welcome to the show.
Hi, Robert. Thank you very much for having me. It's always a pleasure to talk with you about health and longevity.
It's good to meet you, Aronica and Lucia. And I'd like to start off by just telling the audience how you got interested in this area.
Nice to meet you, Stefan. Yes, I am Italian. I was born in Italy, and I started my career in epigenetics in Vienna when I pursued my doctorate studies. And epigenetics for me provided a new way of looking at food. After studying epigenetics, so I had being born in Italy, I always had a strong relationship with the food, and food was part of our tradition and approach to health, both physical health and emotional health, because we used to eat with family and friends. So food has always been a very important part of my approach to health since the time I was born. But when I started my career in epigenetics, this provided me a new window, a new perspective on the relationship between food and health. And I realized that food is one of the most potent signals to our genes. Food is not only calories, but it's information. It's information that our cells use to reprogram the genetic software, the program of genes that we turn on and off in our cells. And this is what makes us who we are every time and who we become. So this software program of epigenetics can explain, for example, why when we change diet or we change an exercise program, we become different. We lose weight or we become stronger. And that's because these lifestyle signals, including food, send signals to our genome and reprogram our genes accordingly.
Well, epigenetics is certainly in the news today, and there's so much going on with it. All the way from first these biological clocks that maybe we'll talk about later, and that are based on epigenetic DNA methylation patterns, but also, as you mentioned, epigenetic reprogramming, either through food or lifestyle, or even OSK Yamanaka factors. But before we talk about any of that, maybe you could set the stage and just introduce our listeners of what is the epigenome versus the genome, and how does it work?
Yes. So the genome is our DNA, the DNA in our cells, which contains the information to produce the proteins that make up our cells. And so the genetics describe really the sequence of the DNA. The epigenome, epi means, the prefix epi means on the top, from the Greek language. So on the top of our DNA, of our genome, there is a second genome, a layer, the epigenome, made of molecular switches that can turn genes on and off. just like a dimmer switch modulates lights up and down in the room. So the epigenome is this set of molecular tags that are placed either on the chromatin, so either on the DNA itself, on cytosines inside of our DNA, or on the histones. Histones are proteins that pack the DNA inside our cells to make it compact and to organize it in a way that our cells always know where to find the genes that needs to be activated or deactivated at any given time. So histones plus DNA coiled together makes the chromatin. And the chromatin is the gateway to gene expression. And the epigenome is a set of molecular tags on the chromatin that regulate gene expression by either closing the chromatin. So when the chromatin gets compact, it's in a closed conformation and genes are turned off, or by opening the chromatin. So the chromatin gets open, the gene can be read by the cellular machinery, and so these can activate genes. So all this magic, this set of molecular tags that regulate the opening and closing of chromatin and thereby gene expression is the epigenome.
So just to clarify, that sounds like it's an all or nothing, on or off, not on a continuum, correct?
No, this is a very good question. It's in a continuum, actually. Sometimes when we explain the rules, we simplify things, but the reality is that it's more a modulation. So you can, we say, in a scientific terminology, we use the word down-regulating or up-regulating, which indicates more a modulation of gene expression up or down, rather than turning off or on. So yes, things are modulated in up and down, and in time. Sometimes you need to modulate up the expression during a certain window of development during life, and then down-regulated later in life. So it's time, space, which cells, and quantity.
And can you relate this to mechanisms of aging, this process that you just described?
Yes, actually there are many studies. Just a recent one published last week by the group of Professor Devin Sinclair, showing that the epigenome is one of the main mechanisms of aging, and probably according to this theory, the aging driven by epigenetic mechanisms is associated with a loss of information, of so like a loosening of this process of modulating things up and down, so that the wrong genes are upregulated or downregulated in the wrong cell at the wrong time, so that the cell lose information and their identity. So what I didn't mention is that the epigenome and epigenetics is crucial to cell identity. And why is this the case? Because we have the same DNA in every single cell of our body. And yet, our cells all look different from each other. Why? It's because of the epigenome. So this tight regulation of gene expression that make, for example, our fat cells express different genes than our brain cells, so that they look different from each other. Now, when this process loosens up with aging, or it's not as tightly controlled, then there is a loss of information. There is a fat cells that it's almost starting to lose its identity. It's not completely, but there's some losing of information. And so this is the theory that this loss of epigenetic information is associated with aging. And according to the last publications in the field, it seems that if we reprogram the epigenome, so by fixing this epigenetic drifting with aging, we can rejuvenate cells. So yes, epigenetics is one of the 9 hallmarks of aging. I mean, there are probably 12 or more according to the different theories, but it's one of the fundamental mechanism of aging, and it's basically the software of our cells, and that's why we talk about epigenetic reprogramming when we describe how nutrition can program the epigenome especially during early life, when the epigenome is very sensitive, or we talk about epigenetic reprogramming, where we use Yamanaka factors in the lab to reprogram the epigenome to rejuvenate its state to a younger biological state.
So it sounds like there are 2 distinct types of or areas of programming going on with epigenetics. So in other words, one is from the primordial stem cell that allows the epigenome to be reprogrammed to differentiate into a liver cell or a skin cell or that sort of thing. And then once that's done, beyond that, there is further epigenetic modification that, at least according to the epigenetic theory, information theory of aging, that actually drives aging as well. So if we want to rewind, reprogram, we want to reprogram back only the second group, but probably not the first group unless we're making stem cells in the lab, right?
Exactly. I like this summary that you gave, and I used to classify the epigenetic modification in those written with a pen that we cannot change, and usually these are the ones in the first category you mentioned. So the ones that are established very early in life and are crucial to cell identity, and that's for a good reason, right? We don't want to erase that pen marks. And then there are the pencil marks that are potentially modifiable through lifestyle. But I think now what we are trying to do is also to potentially erase possibly with new drugs, even some of the pen marks that may be associated with aging, and that we acquire either early in life. So these are not the ones that are essential for cell identity, but we do acquire early in life some disease associated marks, and some of them may be potentially written in pen. And what we hope is that the new technologies in epigenetics may help us even reverse those acquired pen marks that are associated with disease or aging. So, but this is we still don't know whether this is possible, although the first there are data suggesting that this might be possible.
when Yamanaka did his initial Nobel Prize winning work with the four Yamanaka factors, wasn't his goal to reprogram differentiated cells into pluripotential stem cells? And that was sort of the whole goal, right?
Yes, and yeah, and just to provide again a summary of that, what happens usually during fertilization of an egg and sperm. So the egg and sperm are very specialized cells with a very specialized epigenome, but then when they fuse together, most of the epigenetic marks are erased, and in the embryo, by erasing and stripping these epigenetic marks, we create embryonic stem cells, which can become any type of cells because they don't have epigenetic marks. So the Yamanaka factor a little bit mimic what is naturally happening during fertilization of an egg and sperm together. Yes, so this is, and interestingly, when this happens, also the biological age, so we can now measure, and I'm sure you and Stefan have talked about that epigenetics is also a way of measuring our biological age. And when during fertilization, or also after administration of Yamanaka factors in the lab, we reset the biological age of the cells to basically zero. And so there is an epigenetic rejuvenation by doing that. One thing I would like to mention, and I think is not been discussed very much in podcasts or in the mainstream media, is that I mentioned that during fertilization, most of the epigenetic marks are erased, but not all the marks. There are these imprinted loci. Imprinted means they maintain the epigenetic marks either from the mom or from dad. So they are maternally and paternally imprinted genes and loci in our genome. And a few months ago, the imprintome has been finally published. I mean, we know where these loci are, and this has been a landmark achievement in the field. Because it's a very new discovery, for now we only have a great tool and to formulate hypotheses and test them, but we still don't know. And why I'm mentioning this, because some scientists in the imprintome field, and I've spoken with Professor Randy Giotto. He's the professor who did the experiment with the agouti mice that I will, if you want, I will describe later, but it's one of the landmark experiments in nutritional epigenetics. So he has been involved in publishing the imprintome, and he believes that many of these imprinted loci are going to play a role also in longevity. Again, this is just an hypothesis, but it's a very interesting hypothesis, because for these imprinted loci, so their expression is more carefully regulated than all the other regions of our genome. So why? Because we always have a silent copy, either from mom or either from dad, for good reasons. And whenever we have a tiny imbalance in the expression of the paternal or maternal allele, we usually see some alterations in behavior or metabolic health. There are some syndromes associated with imprinted loci, and many psychiatry disorders have been associated with that. And some conditions are not even compatible with life, like, if many miscarriages may be due to the alterations in imprinting. But my point is that, I think we are classifying now the epigenetic modifications in pencil marks and pen marks or categories, but the epigenome is so complicated that we are just scratching the surface. I'm very excited for the future of epigenetics, and especially with the imprintome research that has just been made available. And Robert, you may consider interviewing Professor Randy Giotto if you want in one of your next podcast, and I can make an intro. He's a wonderful researcher with lots of visionary ideas for the field.
Yeah. This is a fascinating area, Lucia, because from a psychological perspective, one of the questions is environmental effects. Which ones of those, how are they, or are they transmitted from generation to generation, such as trauma? And what I hear you saying is that there is a mechanism by which some of life experience, aside from nutrition, some of life experience does get passed on from generation to generation.
Yes, so the topic of transgenerational epigenetic inheritance is a controversial one. So we know that it happens. It happens. We don't know how. So what I mean by that is that there are two ways by which transgenerational epigenetic inheritance can happen. It has been demonstrated in plants and worms, but there are two main ways, either through what is called intergenerational inheritance or through transgenerational inheritance. What I mean by that, the transgenerational inheritance really happens through the male or female germline, which means I need to have some epimutations. So epigenetic modifications written in pen that don't change and are transmitted through the sperm or the egg. Or the intergenerational inheritance is due to direct effects of the environment in utero on the germline of the baby. What I mean by that, the egg that made me was already in my grandmother. When my grandmother had my mother in utero, her diet was affecting directly the egg from which I was born. So what I mean by that, this is if my grandmother was eating a poor diet or something that, that might have affected me directly, not through her germline, just because her nutrition was affecting my epigenetics. This is still an epigenetic inheritance, but is an intergenerational, right? Because we don't, especially for the female line, we cannot separate that until we come to the next generation. So let's say that I have something, a trait that I've seen in my grandmother, and like a metabolic issue associated with a certain epigenetic change, right? There's this association in my grandmother, in my mother, and in me. I still don't know whether this is intergenerational or transgenerational. Only if my daughter still has it, then I can say, okay, then my daughter was not exposed to my grandmother's nutrition whatsoever. Mm-hmm. It must have been a mutation in the germline. So this is just to, again, to provide you with another layer of complexity, understanding why scientists don't agree and say like, we have not demonstrated that. There are some observations that the transgenerational inheritance is happening. We know, for example, the Dutch hunger winter is a famous example of that, where there was a famine and the baby born during the famine were born underweight, but then in adult life, they had a higher risk of developing metabolic disease. And after 6 decades after the famine, when they had plenty of food available, they still had some epi mutations in metabolic genes crucial for glucose metabolism and lipid beta. So this is a candidate hypothesis for a possible transgenerational inheritance, because also their babies were never exposed to the famine, had the same epigenetic mutations and a higher predisposition to metabolic disease. And usually they were born underweight. So this is still not demonstrated, but it's an association. So, and we have plenty of other examples of associations with trauma, even from the 9/11 survivors. There were some epigenetic changes associated with the behavioral genes. We have even for war, the survivors from the war, we have epigenetic changes, but it's still not demonstrated. Right? It's still, we don't know the mechanisms, but there are many indications. And one last example, in the field of metabolic disease, we know that the sperm cells of obese men have obesity-associated epigenetic modifications that are different from the sperm cells of lean men, pointing to the possibility that obesity, the gaining weight and the epigenetic consequences of that might be passed to future generations, because if the sperm epigenome is changed, then there is a higher chance that this might be passed to future generations. So there are so many hints. Again, scientists don't fully agree on the mechanisms, but there is plenty of evidence in animals. In animals, unfortunately, we have evidence of transgenerational inheritance. I say unfortunately because most of this evidence is associated to exposure to toxicants that we are also exposed to, including plastics, glyphosate, and jet fuel and other stuff. So, and unfortunately, in mice, we have evidence that those exposures induce transgenerational inheritance up to the 4th generation. And it's easier to do these experiments with mice, first of all, because we don't have ethics problem to exposing people to this bad stuff and see what happens. And then second, because we can follow them through many generations and establish causality. So unfortunately, it seems, yeah.
It's such a fascinating question. It reminds me of a book that someone wrote called "You Are What Your Grandparents Ate." But this question between intergenerational or transgenerational epigenetics, as you mentioned, some of the older, like the well-known Dutch hunger study or the Holocaust victims study and all those, seems like those would be far enough out that you could get beyond 3 generations to answer that point. Have people looked at that? Do these traits extend beyond 3 generations, or just they don't have the data yet?
They don't have the data yet. As far as I know, I didn't see any study. In this case, it should be the grandchildren. For the female line, we need the third generation. So in this case, for the male line, men are more simple, right? You just need to go to the third generation. That will be enough, because the sperm formation is affected during adolescence, whereas the eggs forms already in the womb. So that's the difference between men and female. So actually you need just to follow up to the third generation in the female. And so it will be for the Dutch Winter, the children, for the mother, yeah, would be then the children of the children of the famine babies. So I think we're not, I'm sure there is a follow-up there. Everyone wants to look at their epigenome. I'm sure that the publication will come soon. And so there will be not so much debating about that.
So Lucia, what is your approach to using nutrition to help in this process of either healing epigenetic wounding, or just to making it healthier? Even how do you describe that?
Yeah, thank you very much. I think that's a very important question. I describe, and this is a word I made up. I made up a word called epi-wellness, which is
good word.
It's more, it's a framework of thinking about all our exposures in terms of epigenetic consequences that they may have. So it's beyond nutrition and includes exercise, toxic exposures, and so on. So epi-wellness is really a knowledge-driven and proactive attitude to health, where we think about the effects of our lifestyle exposures to our genes. And in terms of nutrition, particularly, I talk about epinutrients, nutrients that are necessary for the proper functioning of our epigenome. And these are some of the nutrients that have been tested in nutrition studies, showing that without their presence, there were epigenetic consequences, especially in mice, in the progeny of the mother that had a diet poor in epinutrients. So what are these epinutrients? Epinutrients are broadly classified into two main categories: methyl donating nutrients, or methyl donors, which donate the methyl group, the building blocks for methylation reactions. So DNA methylation is a kind of epigenetic modification, and methylation of histones and even RNA molecules is another epigenetic modification. So we need methyl group to power epigenetic reactions. Methylation goes beyond epigenetics. It's important for many functions, including DNA synthesis, immune function, but it's a key process in epigenetics. The second category are so-called epibioactives. So nutrients or phytochemicals in the food we eat that can modulate the activity of epigenetic enzymes that write, erase, or read epigenetic modifications. And these epibioactives are almost everywhere in the whole food we eat, in the plant, especially plants. So, for example, tea catechins, coffee, resveratrol is one of those epibioactives. Although, despite the controversy, I'm not suggesting you should be taking resveratrol. I'm just making the point that resveratrol modulates the activity of erasers enzyme in our cells. And in broccoli, we have sulforaphane, a very popular epinutrient, epibioactive, and the list goes on and on. The point that I always make is this one. We tend to focus on on a battle between, you know, low carb, low fat, or vegan, carnivore. But there is a one, I think we can all agree that if we eat whole food, nutrient-dense food, we maximize the nutrients we get, and I'm talking here about epinutrients, right? They are rich in low glycemic foods, especially because low glycemic foods and fruits and vegetables have fewer calories and more content of nutrients and epinutrients. And and I think, you know, if we really focus on a whole food diet, we are automatically going to have a lower carbohydrate diet because we don't eat refined carbohydrates and sugar, and a higher epinutrient diets. And I think we all agree on that. And then, you know, and then I think this should be a universal base. Then for some people, they need to restrict carbohydrates more because we can use the macronutrient composition of the diet as a therapy. There is for example, a ketogenic diet that can be a therapy for certain conditions, both metabolic conditions, psychiatric conditions. But again, this is the 20%, or we need to avoid some foods because we are intolerant. We may be intolerant to lactose or to some specific vegetables. There are people who are intolerant to nightshades. But again, like, the framework should be whole food, which and epinutrients, building blocks for our epigenome when we eat, and the flavor, and also re-educate our taste buds to capture the variety of flavors in these epinutrients. Sometimes they are bitter, sometimes, and we lose the taste and for these nutrients that are so good for us.
Yeah, I think that's a great message about whole foods, nutrient-rich diets, avoiding processed junk foods. I think everyone would agree with that, and it's such a great message, and so many great messages here on this program today. How can people reach you? I know you have a course coming up that you're teaching that's going to be available, and how can people reach you on social media or your website, Lucia?
Thank you very much. So I do have a small social media presence. I have a YouTube channel where I share my Stanford lectures with longevity scientists, epigenetic scientists. So I invite you to check out my YouTube channel and subscribe. I am on social media, on Instagram, Dr. Lucia Aronica. And I'm about to launch an epigenetic course, very compact for professionals, so health professionals, but also industry experts, administrators who want to know really the basics of epigenetics, the essentials of epigenetics, to give their career a competitive edge in this rapidly growing field. And I will use, I will make available the content I have created for the first ever professional course in epigenetics at Stanford. This is part of the Stanford Genomics certificate currently, but I will focus in this mini course on nutritional epigenetics. So every professional can get started, and I will announce that on my social media and newsletter if you sign up to my newsletter at draronica.com. Very
good. We appreciate that, and thank you so much for this very informative talk. And I hope we have the opportunity to have you on again, and you can tell us about the progress that you and your studies have been making.
Thank you very much, Stefan. It has been a pleasure to know you for the first time and see Robert again.
Thank you.
This is for general information and educational purposes only, and it's not intended to constitute or substitute for medical advice or counseling, the practice of medicine, or the provision of healthcare, or diagnosis, or treatment, or the creation of a physician-patient or clinical relationship. The use of this information is at their own user's risk. If you find this to be on the value, please hit that like button to subscribe to support the work that we do on this channel. And we take your suggestions and advice very seriously, so please let us know what you'd like to see on this channel. Thanks for watching, and we hope to see you next time.